So we just finished talking about genetic linkage in the introduction. Now we're going to move to genetic mapping, and at the end we'll talk about 3-point crosses, and we will do one 3-point cross in the lab. I want that you remember that when we make a genetic map, that the degree of linkage is measured by the frequency of recombination. I want that you remember that the frequency of recombination is the same for cis and trans, or coupling and repulsion, and I will explain in the following slide what is that. And also I want that you remember that the frequency of recombination differ from one gene pair to the next. Okay, so these three concepts are very important for you to know, for you to remember after this section that we talk about genetic mapping. Let's start talking about cis and trans configurations, and maybe from general culture or from chemistry, you remember that cis means the same and trans means different. So in genetics, this concept is also used here when you have the two chromosomes, two homologous chromosomes, and here this is the example of your textbook of the cis or coupling. In cis, you have both A and B on the same chromosome, and here this could be the wild type, and here you have the mutant. Let's put a mutant. They will be on the same chromosome. Then after meiosis, if this is a cis configuration, these two, the parentals, will be on the same chromosome. Yeah. So this will be the wild type and this will be the mutants. On the other hand, the recombinants, you will find a combination between the A from the wild type and B, and A from the mutant and B from the wild type. So to summarize, remember cis configuration or coupled configuration. The cis means that both mutant alleles and wild type alleles are on the same chromosome. So all the mutants will be in the same chromosome and the wild type on the same chromosome, and they will be then the nomenclature. They can be written like this. So here they will be the two that are the wild type, and slash the two that it will be the mutants. Okay, now let's move to the other example, that is the transconfiguration. So now we're going to move into this figure, and I want that you observe and you try to see the difference. Okay, so first thing that you have to observe is that when you have a transconfiguration or repulsion, here you will have a capital here plus a lowercase, and in the other case the same thing. So lowercase and capital, in this case it will be a combination between the wild type and the mutants. Then after meiosis, you remain with the combination in the two chromosomes, capital and lowercase and capital, yeah, wild type and mutant. And then in the recombinants, you will have here the capital, here we consider that as wild type, and here you will have the mutants. So it's completely opposite to the cis if you compare them. So the trans configuration of repulsion, mutants allele are on different homolog of the same chromosome, and they are written in a different way. So here you will have still this slash here, but here you will have a capital on lowercase determine the alleles, and here the same thing. So again, they are the opposite concept in here cis and trans configuration, or coupling and repulsion. Now we're going to integrate different topics that we discuss in the class and also in the lab. So in the beginning, we're going to this concept of linked genes. We're going to integrate it with these cis and trans configurations that you can see here. And then in addition to that, I want that you think that in the context of course, fruit flies. And Maybe we already have worked with these fruit flies. For example, this mutant, we already talked about that, and possibly in the lab we already worked with that. And we're going to make this cross. Female and a male. We're going to cross them, but this female has a white eye, so the mutant symbol here we can put a W. And if you see the wings will be wild type. So generally the symbol for wild type can be plus. Now I want that you see what is happening with the eye here and with the wing. So here the eyes will be wild type, so of course we will have as a symbol plus. And here the wings are miniature wings, so we're going to have it's a mutation, so we're going to have an M. And now after we cross them, the progeny that we can get, it could be different. We can have a trans or a cis configuration. So let me show you an example. Let's start with A that is trans. So as we said, here we have one wild type here and the other one wild type here. And they are in different chromosomes, so they will be considered trans. Here you have the white eye, and here you have the miniature wing. So they will be written like this, as is showing this schematic with this symbol. And I wanted to introduce you to my cat. He's meowing a little bit. Hopefully you enjoyed, and he might be start getting vocal but let's keep talking about the genetics. So now let's go to the other example. It's a Cis configuration, and if you see both mutants, the white and the miniature here are on the blue chromosome, while here in the green chromosome you find the 2+. And if you work in a lab or if you read a scientific paper using fruit flies, you will find that there are different ways to scientifically communicate what are the genotype of the fruit flies, of your fruit flies. So there, in addition to this one, some papers can use this line here, but many of them they just use a slash. Why? Because it's easier to write it in a paper with a slash. And in some cases, so for example, you can have the white like this, but you can also have the wild type as a plus, or you can have the gene as an M plus. So it means that is the wild type version of the miniature wing. So it will be a wild type wing. And the same thing happened here. You have the M, the mutation for both, and here you will have the wild type for the white eye, or it can be right like W plus. So they are just a different type of writer that they have the same meaning, and in this way is also the same here. It can be right like this, and with the plus or with the M plus, and it means that they are the wild type. And if you read a paper or you have any doubt, there is so many things that we can learn about what are the type of symbols that we use when we talk about fruit flies. And later in the class we will talk more about that. So remember, in those two cases, this will be an example of transconfiguration, while this one will be an example of cis configuration. So later we will go back into the repulsion and coupling, but now to do that we first have to go into this topic that is genetic mapping. And what is genetic mapping? A gene mapping describes the methods used to identify the locus of a gene at the distance between genes. So we know that here there is a chromosome. Now we need to start looking what type of genes are there, and the way that they start doing that, it was using mutations. So here, so for example, well, generally in the wild type, for example, you have this type of antenna. They found mutants that they have tiny antennas, while Um, the wings tend to be flat because they are used for flying. They found that you can have flies that they can have different morphologies. They can be bent, they can be smaller, that they are not useful for flying, but they found that it could be in the similar chromosomes. And there is a hundreds and hundreds of mutations that can be useful for mapping the chromosomes. And in the lab and also in the following slides, I will give you some examples. So here is the cytogenetic map. Here is a genetic map, and here there is a physical map, and now we have a DNA sequence. And all in theory are relatively similar, but they are not the same. Yeah. So the genetic map are the ones that I will show you today. They were created by Morgan, and they are created in based on frequency of recombination. The cytogenetic maps are used with stainings, but they don't give you exactly, they don't tell you exactly which gene are or where the gene is located. And the physical maps are the ones that we have currently, that we are using DNA sequence to start to tell you where a gene starts, where is the promoter, where is the last part of the gene. And however, it's very interesting that the first step to produce these physical maps were based on frequencies of recombination, what is unbelievable, and I think it was a very incredible idea. So uh, genetic mapping was used using recombination frequencies between allele in order to determine the distance between them. So the closer that they will have a lower frequency of recombination. The farther, they will have a higher recombination frequency, up to 50%, as we talked in the previous section of the lecture. As the distance between genes on the chromosome increases, so the fraction of recombination, as I just said in a second. So here, when you have a small distance, you will have a small frequency of recombination, and when you have a large distance, you will have a larger frequency of recombination. And we will talk about the distance, the different type of measurements. In the case of the genetic maps, was map units, and this is equivalent to 1% of recombination, or 1 centimorgan. And we will talk in the following slides about different ways to measure this distance of recombination. So now, to talk about mapping, we also need to talk about the different units that are used. So I just remind you that in the past we used this fraction of recombinants, and they found that this fraction of recombinants reflects the distance between genes. And so in the past we have multiple units that they were mainly based on this percentage of recombinant. One of these was called map units, and in Drosophila they are often called centimorgans, that due to Morgan, and they go, as I said before, from 0 to 50. So from 0 to 50, 0 to 50 centimorgans. And again, this was not the physical map of a chromosome, but it gave us the idea between genes before the gene sequence was invented. Now the sequence, the physical maps are measured in base pairs. And for example, 1.3 centimorgan is around 1 million bases in humans. And this way of measuring distance between chromosomes, it allows us to determine the linear order of genes, so where the genes were located, even we didn't have the sequence. So remember, the genetic map depends in the beginning of the recombination result from crossing over that we have been talking, and we use some different markers to determine where are the genes, and the linkage of genes can be represented as a genetic map. and this will give us a linear order of the genes along the chromosome and space, and the distance between genes is proportional to the frequency of recombination between them. So to finish this section and do some exercises, I just remind you that recombination can be expressed in 4 equivalent ways. This is in your textbook. There are a few questions about that, and the questions are very easy. What they are just saying is, imagine that you have the mutant number 1, the fry mutant number 1, and that is a female, and you have a cross with a male that is a mutant number 2, and then you got a progeny that it will give you 1,000 flies. And those 1,000 flies will be, some of them will give you the genotype of the parental, and they will give you the genotype of the recombinants. And we will show you that with the schematics in the following slides. And in this case, so for example, you got 19 flies that look like a recombinant and 12 flies that look like a parental. So you add 19 plus 12 and you will get 31. Good. So those recombinants, you will calculate that and you will say that the recombinant type will be 31 divided 1,000 is 3.1%. The genetic distance is the frequency of recombination, and it will be 0.031. The percentage of recombination will be 3.1. Another way to measure that is with map units, as we said before, and in this case will be 3.1. And if we map the distance in centimorgans, will be 3.1 centimorgans. So again, this is not very difficult. There will be some questions related to this, and this, of course, it will be much clearer in the following slides when I show you an example between a cross with fruit flies and the parentals and the recombinants. But I just wanted to tell you that there are 4 equivalent ways to measure recombination, and you will see it in a second. So now we're going to talk about mapping, and we are going to talk about this in a historical perspective. So they use fruit flies, and to do that, they use X-linked genes. And if you remember the previous chapter and also genetic revolution, this guy is fundamental. Thomas Morgan, and he helped us to establish fruit fly as a model organism to study genetics. And in addition to study the wild-type mutant, he studied several X-linked genes that they were great tools to study linkage. So let me briefly show you what he did. So again, we have the 2 fruit flies that we talked in the previous slide, wild types that they have the white mutants, but they were wild type for the wing size. On the other hand, we also had these males that they were wild type, as you can see here with this plus, for the eye color, but if you see here, they were mutant for the wing size. And just a reminder, well, these were here homozygous because they have 2 X chromosomes, so they will be homozygous for the white-eyed. Yeah, so they were pure lines. Here, this will be a hemizygous. Why this is a hemizygous? And I remind you that males in fruit flies only have 1 X chromosome. So far here, the crosses will be normal, and in the F1, what they did is to set up a very interesting cross. We set up crosses when you have double mutants. So for example, here, this will be a test cross. You have the 2 mutants here, as you can see here. They will be again hemizygous. And also you will have this line, the females, that it will be heterozygous for both the wild type, for the white eye, and also for the miniature eye. So, and I remind you, these look like a dihybrid cross. So these will, the ratios that we were expecting, it will be something like a 9331 or something in similar to that. And what they found was something that it was very different from that. So here you can see what happened in the F2. Here you can see the F2, and I want that you take time to see what do you see here. What happened between the two ratios? Do we get a ratio that is 1, 1, 1, 1, 9, 3, 3, 1? What is the ratio that we get? And I want that you think about it. Okay, now that you have time to see the graph, now I'm going to show you that this is an schematic shown in your textbook. They took 60, when you add all these flies, there were almost 650 flies. And the interesting thing is that you have two groups. Here you see the one group that they will have the parental types. And these are almost 77%. So one of them will look like they will have white type eyes, and the other ones will have the miniature wings. Yeah, so most of them will look like that. However, you also have almost 34% of those ones that they will have recombinant types. Yeah, recombinant types. So, and here you have the two type of recombinants. So recombinants that they will have, or this one, the flies will be completely wild type. They will have no mutants, or they will be double mutants. Yeah, so they will express the white eyes or the miniature wings. So that was fascinating. And the question is, okay, what has happened with these flies? Why they display these very weird ratios? And I want that you remember this, that we will be covering in the following slides, that the degree of linkage is measured by the frequency of recombination. So this relationship between the parentals and the relationship between the recombinants will tell us the frequency of recombination. And I will show you that in the following slide. So now, before I explain how to calculate the frequency of recombination, now I want to show you this cross. In this case, again, we have the wild type, sorry, the white-eyed females, and we have a male that is a mutant, but in this case it's a yellow body. We also work with these fruit flies, with this type of mutant. Now the question that I have for you is, could you please, before that I explain, could you show me how do you think that it will be the F1 and what will be the F2? Of course, I'm not asking you to calculate the recombination. I will give you that data later, but I want that you set up the crosses that you think that it will happen. Okay, what will be the tester cross? What will be the double mutant? What will be the heterozygous? You can stop the video, you can go backwards. Please calculate, show me. There is no need to calculate, but try to draw what will be the parental lines here, what will be the recombinants here. And in a second, I will show you the answers. Okay, so this will be the first answer. In this case, this will be a trans repulsion heterozygous. Yeah, but here you can see it's heterozygous. And here you will have, again, this double mutant that is a hemizygous. And now I'm going to show you the parentals, and I'm going to show you the recombinants. So these will be the parentals. Again, the parentals will look similar to the parents, so they will be wild type body, but they will have white eyes, and the other one will have yellow body but red eyes, while the recombinants will be completely wild type, or they will be double mutant. Yeah, double mutant, that they will have yellow eyes and white body. So now let's study this in more detail. So remember, now we're far away from the ratio 1:1 that we were expecting. Why? Because this is a test cross. Yes, we are having, if you see the tester line here that we have here, this is the recessive lines, and here we have the heterozygous. So we were expecting to have a ratio 1:1:1 if this was if there was no recombination and there were just two genes and each one with two alleles. We were expecting that we will have 1:1:1 ratio. However, here in this case, it's even more dramatic, the difference. We have 90.6 or almost 99, and here you only have almost 1% of those changes. And this is telling us something very important about how close or how far are those genes from each other. So again, remember, these are far from the ratio 1:1:1 that we were expecting if we were following the independent assortment. And how we can calculate this 98.6%? Very easy. You add the two parentals. So this 4,000 and this 4,600, and they will add, and you divide it by the total of flies. So here you will get this value, and this will give you a percentage of 98.6%. And with the recombinants, you will do something similar. If you want, you can start calculating the recombinants. Okay, so now the recombinants we will do something very similar. So here we will add the two recombinants, 86 plus 44, and we will have a total of 9,000. So you will get something that it will be around 1.4%. And this number, why this number is important? Because the recombination frequency increases with increasing distance between genes. So these one are very close to each other, even more than the white eye, the gene white eye and the miniature. And no matter how apart those genes may be, the maximum frequency of recombination is between two genes will be 50%, that it will be the normal amount between independent assortment. And I show you the examples using genetic markers that that we use fruit flies, but now we can also use PCRs and different gels that will allow us to see the same behaviors. Just that this was used by Morgan a long time ago, but the same logic still take place when you use more molecular techniques. Now let's add an additional layer of complexity, and now we're going to talk about the frequency of recombination. And we are going to talking about the frequency of recombination, but with the Cis and Trans Configuration, or also called coupling and repulsion. And we're going to show you that is the same. And so first thing that I want to remind you, all the examples that I have been talking are mainly this type that give you a trans configuration. Yeah, so where you have the heterozygous, that you have the plus, the wild type, and the mutant in one chromosome, and the mutant and the wild type allele in the other chromosome. And how we get that? Because we have a single mutant here and a single mutant here. But so a single mutant for example, white-eyed, and in this case, a single mutant for the yellow eye. Yeah, but the other trait is wild type, as you can see here. So as a result of that, you are going to get a transconfiguration, and either way, you are going to need to cross them with a male that have to be a tester line. Now, if we want to get a cis configuration, it's very easy. You have to do the following cross. So the following cross is you're going to have a double mutant instead of a single one. So you will have a white eye, but also a yellow body for these females. And as you can see, you have here a pure line for both traits. For the yellow body or for the white eye, they will be homozygous. While here you will have a wild type males, and this wild type male for both you see them, then they have red eyes and a normal size wings, and as a result you're going to obtain this wild type in one chromosome and mutants in another chromosome. Either way, you will need to have the tester line that you should be a double mutant, and males should always be there for the tester line, remember. And here is the example that you should get. Yeah, these are just how the chromosome will look like. Yeah. This will be the repulsion or trans, and this will be the cis. So now the next question that I wanted to ask you is, could you please stop the video and try to tell me which will be the parentals? Well, for the F2, I want that you find the F2, and I want that you tell me the parentals and the recombinants for this one and the F2, and I want that you tell me the parentals and the recombinants. Again, you don't need to have the data perfectly. I just want that you write the genotypes. Okay, so now let's see the parentals of these flies. So here you can see again that here there will be the parentals, if you see them here. So the highest proportion will be again the parentals, and you can see these data points that they are very high, 90.86, and here you can see this, that they will be the recombinants, that they will be in a much lower proportion. Now the interesting thing is when you do the cis configuration, you will find that the parentals right now, they will be different. They will be the double mutant and the wild type flies, and those ones will be now here. So this one will be your parentals, and this one again have a high frequency, although they before here they were the recombinants of this core of the previous cross. Now in this case they will be the parentals, and this is why they have such a high frequency. On the other hand, here the single mutants, here they will be the recombinants, and as a result of that they will have a much lower frequency. However, independent, as I said, of the crosses, of if you have a configuration, a coupling or repulsion, you are going to get the same frequency. So this one is 90.86, this one is 9.8, so it's very close. And why is not exactly the same? Because you will need to have a really high number. Actually, this is very close, and these are estimates. And here you will have the recombinants will be 1.4, and here you have 1.2. Again, very close. So it shows that the frequency of recombination between the gene white and the gene yellow, they are very close to each other. Okay. Now let's try to do another example, and again you have to add the white, sorry, the F1, the F2 for both cis coupling or the trans repulsion. Okay. Okay. So here, remember, we have ones that have only a single mutant. Here you have one that is also a single mutant, and here you have a double mutant, and here you have the wild type. So please try to tell me which one is the F1. Try to write which one are the repulsion, which one are the coupling, which one are the configurations, and then please go again and find the F2, which one are the parentals, and which one are the recombinants. Again, I haven't give you any of the data points. I just want that you make the crosses and try to determine those lines. Okay, so let's start with the F1. And so you can stop the video if you don't want to know the answer yet. But let me show you the F1. So here you have, here you will have the repulsion. Yeah, very easy, very similar to the exercise. So here you will have a heterozygous with both mutant and wild type in the same chromosome, and here you will have your tester line with the two mutations. And here you will have the coupling, the Cis configuration, where you have in one chromosome the mutants and in one chromosome the wild types. So that was very easy. Now let's go for the F2 with the parentals and also the recombinants. So here, the parentals here, very easy. Here, the one that are the single mutants, and here, or the wild type, or the double mutant. While here, the parentals will be the opposite. So let me have here a P here for parentals, and here recombinants, and of course this will be the opposite. Here this will be parentals, and here you can find the recombinants. And again, similar to the previous exercise, you can calculate these values. So here you have the two parentals, and you will get a frequency of recombination of 66, and here you will get a one that is very close, 60.3, and also a very similar the frequency of recombinance, 33.5, and here 33.7. And I hope that with this exercise is clear for you that the frequency of recombination is the same when you have a trans configuration or repulsion, or this one, a coupling or a cis configuration in the chromosomes. Now we're going to finish, and if you remember, I told you that the frequency of recombination differs from one gene pair to the next one, and I have been showing you many examples with the white gene. So we have the white gene, and you cross it with a fly with yellow body. So what is the eye color and the body color? Those two genes, we want to see what is the distance. I also show you with the wing size and the white eye, and you can find the distance between those. And here, even with another mutation here that make the body of the flies very small, you can also calculate the distance. And again, you will just do exactly the same type of exercises. And I just want to finish showing you this. Here you have again the parental types. Here you have the the percentage of the parental types, and here you have the measurements that we talked in the beginning. So you have 19 and 21, 19 and 12. So the recombinant times will be 3.1%, and you have all these four ways to represent that distance that are all equivalent, if you remember. All of them will be equivalent. And with this, I finish this section of this lecture. If you have any doubt, you can please go to the textbook. I try to use as many examples from the textbook, but there I think that if you have any doubt, they will explain it in more detail. Have a good day, and we will talk later.